to the new and better way of doing things, which is using molecular marker. And when I say molecular here, it actually means any molecules, any biomolecules in your cell. Everyone loves biochemistry, right? Love. Best subject ever. Ajahn Pim is the best. Right? You hear this, Ajahn Pim, everyone loves you. You just think, yeah, I love chemistry. Right? So biochemistry, it can be anything. Anything from, you can use protein, you can use RNA, you can use DNA for this. Right? So any of this has been used as a marker. And what markers are we'll talk about very soon. But in this class today, I'm going to focus on DNA because that's one of the easiest things to work with the systematic. So let's focus on DNA today. But just so you know, if I'm going to teach this class again in 5 years, I think I might have to start teaching about RNA and proteins too. But that's a subject for another day. Or if you study, if you take, isn't bioinformatics required? No? It's fun, but it's like difficult. But if you take bioinformatics, this will be taught in that as well. So what are DNA markers, right? So markers, is a marker in today? This is a marker, right? You can write stuff with markers, but marker in DNA is a region of the genome. So genomes mean, in the literal translation, is all genes. Or a better translation is all DNA. So in each cell of your body, DNA is so long, you can wrap the world around with DNA, right? But we only need one region, just one part of the gene, or one part of the genome that says something about the organism. So that's a marker. You probably have heard of like this DNA testing, right? You can swap the sheet and tell you how many % of you are Neanderthals and how likely you are to have cancer, that kind of stuff. That's a marker. That's a molecular marker. That's a DNA marker. So for us, me, systematics, what we really care about is this. So if two plants are really similar to each other, only maybe like different colors, so the similar organism should have similar DNA, duh, right? You should have similar DNA to your parents if they really are your parents. Oh. Right? Classic case of like drama. Like, you're not my kids, I adopted you. So if the plants or any animal or species, if they're similar, they should have similar DNA. And of course, if they're different things, they ginger and mung beans, then they should have different DNAs. That's the idea. So, and all the ideas surrounding using DNA marker come from this. You compare, basically, all we do is comparing DNA sequence. So the idea is you comparing DNA sequences. So that's the idea. But this main idea sounds simple enough, but the method that we're going to use to compare the DNA is quite complicated, and we're going to walk you through that for the next class together. So the idea is that instead of using morphological character matrix that we just used before, from that, remember the 0 and 1 that we tried to do the Chandler characters, instead of 0 and 1, we use the ATCG here instead, and we turn morphological character matrix to molecular matrix instead. And with this technology, instead of like 0 and 1, 0 and 1, and 7 characters, we can do like 5,000 characters at once, quickly, right? So the steps are here. So we have to choose. You can't compare everything. I mean, you could in theory, and use a lot. It's gonna use a lot of computing power. And I think there's a master not here in my school. You can take a course on comparing everything in the genome. Use a lot of coding, which is really fun and really frustrating. But for now, you're gonna choose one or more DNA markers. You're not gonna choose everything. You're gonna choose only a few places to look at, and then you can do the web lab, or you can get the data online, and you're gonna practice that 2 or 3 weeks from now. and then you align the sequence, and then you find logic in reconstruction. So the first two steps here are basically what we talked about before, is to find the homologous structure. So instead of like, is my arm homologous to dolphin arms? Like, okay, don't worry about that. Let's find the same place in DNA and compare them. You don't have to worry about homology, right? And then you do the lab to get the data. And the third step is to line up, make sure that homologous structure is in the same place, and finally do the grouping. And in this class today, this is lecture 4, and this will be your lecture 5. Okay, so today we're gonna stop at number 3, and we finish early, we leave early, and we enjoy the sunny day that we have. No, I want the rain back. I mean, like, finally we have the rain, but good God, right? Nakhon Pathom is always extreme, right? When it's sunshine, it's sunshine. When it's rain, it's rain! I was actually driving back from Kanchanaburi yesterday. It was raining the whole time. I was like, really? You can't do things in moderation. All right, and Central Nakhon Pathom is really weird, but anyhow, you guys been there? No, not very exciting. Anyhow, so let's start with the first one. Choose DNA marker. Right? So this is typical marker that we use. I think this indicates you want to do projects on this, and this name probably being not with you right now, but you can see that each group, bacteria, fungi, animal, algae, and plants, barely use the same marker. Like the names are different. And the reason for this is arbitrary, meaning that they have used this and it worked. Let's use that. Really? That's what they do? Yeah, this is science, guys. It's like, oh, we try a bunch of different things, this thing stuck. Like, ICS kind of worked, so let's use that. And we're going to move away from this in the next couple of years, but for now, this is what I like to call the legacy marker. They've been around, people use them a lot, so let's continue working with them. So just like, one of the things I really like, and Mahidol is a funny place for this, you guys are required to wear uniform, right? Well, not obviously not, right? But they're like, wear uniform, unity, Mahidol is so great, right? Like, yeah, let's wear uniform. And like, why do we need uniform to begin with? Anyone remember why we wear uniform? Ever? No? Actually, originally, high school doesn't have uniform until World War II. During Jokon the field marshal, Pibun Songkham, he was like, let's make sure we can tell the student apart from the others, because we have military, we have this and that, so make sure we have uniform, and that's that system. And nobody cared to like question why we still have uniform. Because when Mahidol first started, we actually university for medical school, right? And there's no uniform. And then why do we have uniform now? Who knows, right? So that's like a saving. It's because it works, and there's no reason to change it. But I'm going to tell you why this sort of thing works. And there are 3 main criteria of choose the right marker. Because our time at Mandy is limited, you can't be like, describe a Zambia. Oh, he has gray hair and black hair and have sad eye, droopy eye. So by the time you get to my feet, it'll be like 20 hours describing every part of me, right? And you can like describe a Zambia in 1 sentence. You pick the best feature. The same thing here. You can use all the DNA to describe your organism, but it's too much work. Why don't you pick one that works? So there are 3 things to think about, right? Where they come from, the sources, the substitution rate. This, if you remember your genetic class, this is your mutation rate. How much mutation it occurs. And finally, the component of marker. We'll talk about that very soon. All right, so go back to cell biology. Biology number 1. This is your plant cells. Where can you get your DNA from your cells? What are the sources of DNA here? Where would you? Nucleus, right? Where's the nucleus here? That purple thing, right? So you can get, number 1, you can get DNA from your nucleus. And this is typically called nuclear DNA. or is shortened by nrDNA. Or why else besides nucleus, obvious one, because that's where the chromosomes are, right? But in the cells, there are a couple other places you can also get DNA. Which other places in the cells you can get DNA? Mitochondria. Mitochondria is here, so this is mitochondria. Shortened is either mitoDNA or mtDNA. And one last place you can get your DNA is chloroplast. So chloroplast here, choro mean clean, plast mean body. So this usually say cpDNA for chloroplast, or sometimes just called plastic DNA. Not plastic. Plastic is your bag. Plastic is chloroplast. And if I don't know if it's being covered by one or evolutionary biology, mitochondria and chloroplast used to be independent living organisms as being recruited into eukaryotic cells. Like, hey, let's come work together. It's great. We have nucleus, we have cytoplasms, and you can come produce energy for us, you can photosynthesis for us. And after 1.8 billion years, they forget to leave. They're like, oh, we start eukaryotic cells, so that's why they have their own DNA. And so we have three sources of DNA here: nucleus, and mitochondria, and chloroplast. But when you extract the DNA, right? When you extract DNA from the cells, you can't really separate them. You get everything, and all of this together they call genomic DNA. So when you crush the cell, you get all the DNA. You can't really separate the chloroplast and mitochondria from nucleus unless you separate the organelle first, which is really difficult to do. But overall, when you crush the cell open, you get everything. Okay, so the three sources you can choose from, and choice matter. All right, let's look at first thing, which is the mode of inheritance. How DNA being passed on to the babies. So usually, this is typical things happen in plants or in animals for that matter. So your nucleus DNA, right, come from both mom and dad, right? But mitochondria and chloroplast usually only come from the female side. Why? Because mom is always nicer than dad? No? Because when you reproduce, right, eggs have nucleus and let's say in the mitochondria and I'll say chloroplast here. But when the sperm come to fertilize, everyone remember the sperm structure? The mitochondria is in here and then nucleus is here. So when they fertilize, only the nucleus part come in and then the neck and the tail got lost. So usually the only nucleus get merged into the egg cells and they shake everything off. Mitochondria is like your friend who drop you off at the school. Can you drop me off at school? Okay. And they drive you over and you drop off and bye! Go away. I don't want to be seen with you. Go away. So the mitochondria and the cells get taken off and then only the nucleus of the sperm got in. That's usually the case, right? So that's why you have, right now in your cells, you have your mom mitochondria. You never have your dad mitochondria, right? And then, but you have both mom and dad nucleus. So if you think about nuclear DNA, you get both from mom and dad. So sometimes this is called biparental marker. So this is a marker that if you want to know who your daddy is, you have to use nuclear DNA because that's what you get from your dad. But if you think about mitochondrial DNA, mitochondria you only get from mom, not from dad. So if you study mitochondrial DNA in your cells, you can only relate that back to your mother, not your father. So you're like, I'm your father. What's your proof? Mitochondrial DNA, liar. I only got my mitochondria from my mom. Right, so that's how you can test. And most of the plant angiosperm, the one with the flowers, you do the usual step. You only get you get chloroplast DNA from your mom, not your dad. But if you are gymnosperm, so this would be like Christmas tree or the pine trees, it's the opposite. Dad give you chloroplast, not mom. And some plants, some really weird plants, mom and dad love you both, right? So you get DNA from both mom and dad. So in plants, chloroplasts are funny, but for the most part, if you haven't heard otherwise, most of the organella DNA come from mom's side of that side. So if you're really interested in the paternity test, who's your daddy, then you need to use the nuclear. But if you're interested in something deeper, like where your family come from, your mother's side, you use mitochondria or chloroplast. So that's number one consideration. All right? Number two, substitution rate, right? We talked about substitution before, that is actually mutation, but more specifically, this is what we call point mutation. It's a change of base pair in a particular place. One or two places, you replace one base pair with another, and you probably heard this before, right? So this is mostly review, but for example, you can have the regular sequence of TTC, it translates to AAG, and this usually produces lysine. But if something happened to your last base here, base C, it's got substitute, just like you watch sport, right? There's like substitute, we get this person in and this person out. C out, T is in, and instead of AAG, you get AAA, and give you the same thing as AAGA. It gives you lysine as well. So in this case, anything happen to the protein? No. This is like when your female friend get a haircut. You see what happened? What? Anything look different today? No. I get 3,000 baht haircut. Like, really? Look the same to me. So that's like female haircut. I'm sorry for all the female, but usually like that way. It's like you always look beautiful. That's the good answer, right? So tip for guys: if your girlfriend asks what happened, like do I look different? Like, oh my God, what do? You look great. Don't even try. Just like you look great, right? So in this case, called silent mutation. Is it mutation? Silent, nothing happened. Right? But sometimes when you change the first base here, and instead of you get yuga, it stop codon, it stop producing protein. In this case, we call them nonsense. You stop producing proteins. And in other case, you change the second one, instead of stopping, you actually produce a different amino acid entirely. You get from lysine, you get arginine, or I forget what THR is, but anyhow, you get different one, and this one called missense. You get a different amino acid. So these are three possible ways of mutation happening, right? And the one that really matter, the one that really affect you, not the silent one. The silent is like the same, you got the same result. So we call the silent one the synonymous, meaning it's the same thing. Like duh, you get a haircut, duh, look the same, right? So the same thing. And the other one, molecular people are really not imaginative. They call this non-synonymous, not the same. So the mutation that really matters, that makes a difference, is this one, right? This one is the one that actually make a difference. So when they measure how much mutation happen, what people do, they do this thing called over ds. Not calculus. This is the rate of non-synonymous over synonymous. So they compare non-synonymous with synonymous. So if you have a lot of non-synonymous, meaning you have a lot of mutations. So if you take molecular evolution class, which is not offered here, they will ask you to calculate this dA over ds. Not differential, just the rate. So because this is not such a class, I already calculated for you. So this is the non-synonymous rate in plants, primate, and rodent. So let's look at plant first because I like plants. So in plants, which one have the highest mutation rates? Nuclear, right? So in plant, nuclear have the highest mutation rate, and chloroplast is right in the middle, and mitochondria is sort of like really not active. So if you want something that can detect fast evolution, like things that evolve really fast, which one would you choose? You would want nuclear, right? Because that's have. So this is good for detecting fast evolution. And if you want to detect something very slow, then you use mitochondria. And I'm gonna add here for my own experience, super slow, because mitochondria barely change in plants. It stay fairly constant in plants. So people don't really like to use them very much. It's too slow. So in plants, people like happy medium. They use chloroplast genome because it's sort of like middle range. What about animals? Which one is faster evolving? Mitochondria. So in animals, mtDNA is faster, right? So this is actually very popular for population analysis, like paternity, like family relations, like you belong to which mom and those kind of stuff. If the family is together, mitochondria is really popular. Like it's really fast. It's a lot of mutation. The nucleus is lower. What happened to chloroplast? Do animals have chloroplast? No, right? Not should have chloroplast? That way you don't have to go to cafeteria, you don't have to go to Shabu anymore. If you're hungry right now, Achan, can you turn on the shade so I can photosynthesize? Wouldn't that be great? No? Do you know how much energy you need for photosynthesis to be able to be actually full? A lot. By a lot. So imagine this. Anyone like chocolate? Chocolate, good, right? A bar of chocolate, just one small bar. If you want to eat a bar of chocolate, if you want to get the same energy from a bar of chocolate from photosynthesis, how many hours do you have to stand in the sun to get that amount of energy? More than 12. What? 72 hours. So, is that 2.5 days for one bar of chocolate? By then, I think you probably burn up already. So I'm not talking about 2.5 days, like 2.5 days of sun. So continuous sunlight for 72 hours to get that much energy. So that's why animals don't use chloroplast, right? The energy is too slow. So we don't have chloroplast to worry about. So that's second. And the third one is a bit more complicated. Not all markers are genes. What are genes? I think this will be Ajahn Ping area, right? So genes, come on. What do we mean by genes? Gene is the unit of DNA that produce protein, right? So not all part of DNA produce protein. Actually, very small part of DNA actually becomes genes in eukaryote, like 1% of your DNA actually produce protein, right? So in each gene, okay, let's look at each gene first. In each gene, you have two areas, right? You have exon and intron. Exon is the one that get transcribed into RNA, and then the mRNA get transcribed into protein. So exon is a part that get transcribed and then translate. So both transcribe and translate to get to the protein. So exon is the one that gets jobs done. But intron is a sad one. Intron get transcribed. Yay, I get mRNA, but then you don't get translate. So you're almost there. You're like, this is like good imagine. And the top 10 for Miss Universe is Thailand! And then all people go crazy, right? And then like, the one get Miss Universe is not Thailand. So like, you're the intron basically. So Thailand is forever intron in all the Miss Universe if people care about that kind of stuff. So it's the same thing here. So we have exon and intron, right? So exon gets produced the protein, intron not. And then we have things like this. Intergenic spacer, right? This is mean between, genic is between genes. So this is like a bunch of DNAs that between the genes. People are like, okay, I'm a gene, and then like, that's a gene. So let's say this is a gene, this is a gene, and then there's space between this, but there's still DNA. There's still air between this. What do you do? I don't know. I'm between two genes. Just like your brain, right? There's 2 ears, and this is my brain, just in between. Do nothing here. Nothing happen here. Just between my ears. So that's because, so it just doesn't do anything, just sitting there. And then we have the internal transcribed spacer. When you hear the word spacer, just DNA that has no, I'm reluctant to say this, has no known function. We don't know what they do, they're just there. No known function. They might have some function, but just like, I don't know, you just right there. So in each marker, we can have exon, intron, intrinsic spacer, and internal transcribed spacer. We can have all this in the same marker. So which part among these 4 things, exon, intron, intrinsic spacer, internal transcribed spacer, which part cannot be wrong? They have to be correct all the time, have to be like very strict. The JV code have to be really conserved. Which part? Exon, right? So exon, usually exon is very conserved. Or it's usually the same across organism. You can be wrong because it's a protein, you have to have the right function, otherwise you have malfunction in something. The rest of them, I can be wrong, nobody cares about me. Like if intrinsic spacer is like, I'm gonna change the gene. Anybody care? Who are you? You wanna change? Go ahead. Go ahead. I don't care. Go ahead. I really wanna change, I'm gonna change everything. Okay, sure. No, then. Are you doing protein now? Okay, go ahead, change. So let's look at the real example here. So this is a popular marker in plants. We have ITS, which is the short for internal transcribed spacer. We have 2 of them, ITS1 and ITS2, stuck between 18S RNA, 5' RNA, and 16S RNA. These produce ribosomal RNA. The name should sound familiar to you from Bio 1. What it does, I don't care. So this green stuff coding for RNA. So this green thing, we usually call them a coding region. You can think of it as exon as well, but coding region is more general term. But in this case, the RNA doesn't code for protein. Alright, and then we have the marker, this really popular marker in plants called RBCL. I use it every day, I talk about it every day. In fact, the whole morning this morning I just spent time with RBCL. RBCL is all exon. And RBCL produce one of the most abundant protein on earth. They produce Rubisco. Anyone remember Rubisco? It's in photosynthesis. It Rubisco help fix carbon dioxide in plants. So this is very, very important proteins. Like you cannot miss this. Like RBCL code for the last subunit of this protein. So this protein, all plants have RBCL because all plants photosynthesize one way or the other. And finally, this is popular marker in my group of plants in Bryophyte, TRNF. So tRNL is short for tRNA, is produce transfer RNA for L, which is leucine. You don't have to know this, I just explained this. So TRNL have 2 exons, and in between they have intron. And TRNF, this is producing 3 RNA for phenylalanine. And in between this, we have intergenic spacer. Plant people are weird. We pick the weirdest marker. In animals, we're just like cytochrome oxidase 1 or exon, done. And you're like, oh, I don't like what some of the gray, some of the green. So the green is a coding region, and the gray part is what we call non-coding, the one that doesn't produce protein. All right. Now that you know this, you are on par with my graduate student. Like, okay, guys, I have T3 choice for you in my lab. You can produce data on ITS, RBCL, or TRNF. Which one are you going to use? Okay. So I'm going to walk you through the process right here, right now. So now remember this, sort of. And then let's try answering the following question. Order the following part of DNA by their rate of substitution from the highest to lowest. Wow, this is a game show, right? So this is highest, is a lot of mutation to not a lot of mutation. So we have intron, exon, intergenic spacer. All right. There's sometimes big. Talk to your friends. I'll talk to your godmother. Next week. Which one should have the highest mutation? Not easy, is it? Okay, let's vote. Who thinks intron has the highest mutation? Who thinks exon has the highest mutation? Who thinks intergenic spacer have the highest mutation? Who thinks we should go home now? Okay, that's pretty clear answer. You guys have the highest mutation. So, you X-Men, you have the highest mutation. So of course, this is have the highest mutation, the intergenic spacers, right? Because it is between genes, it doesn't do anything. It don't even get transcribed, right? And intron is sort of medium in most cases, and this one should have the lowest mutation because this exon is have the function, right? So knowing this, which marker should have the highest rate of substitution? All right, I'm going to go back to that structure here. Or maybe I should start with easier question, right? Which one definitely should have the lowest mutation? BCL because it's all exon, right? So we start from low to high here. So the lowest one should be BCL, because it's all exon, right? Have a slow mutation. What about ITS and turn out, turn F? Which one do you think have the higher mutation rates? They both have two grades, right? Really hard. Okay, we're gonna do some board here. The choice of going home now is not an option. Okay, you can choose either turn out F or ITS. Who goes for turn out F? Two, three. Okay. 4. Who go for ITS? 8 minus 4. Okay, thank you. So the rest of you. All right. And drum roll for the answer. This is not on your slide. So this is actually from my actual real data. And you can see that in ITS, this is the spacer. You can see a lot of colors, meaning they have different sequences. And you can see BCL barely have any mutation. And this is also the intergenic spacer. And compare here between ITS and TNF. Which one have more color dots? ITS, right? So ITS actually have higher mutations. But this is a special case because sometimes it's hard to compare this based on the. So in plant, usually ITS have the higher mutations. Oh! My physical therapist say I should focus on my standing. I'm not being mindful and it's on me. All right, so strengthen my belly, strengthen my core muscle, lean back and stand. Okay, sorry. So highest mutation rate goes to ITS. And the last question, all right, so if you want a marker to reflect a recent divergence, something that recently changed, quick, fast evolution, which one would you use? The one with the low mutation or the fast mutation? Fast mutation, you want ITS. Now you know how to set the marker. And let me tell you right now, this is not easy. Like this task that we just walked you through, all the systematics spend years and years discussing, and you can read tons of paper. People are like, I don't think ITS is a good idea. And the people yelling. Actually, I have a friend who's like, really? Use ITS? That's stupid. Why? And there's like a lot of like molecular biology reason behind it. But this is like a basic idea of how people choose different markers for different studies. All right, 5 o'clock. We good time. We good on time. All right. So to summarize everything, right, based on the characters. So let's start from this. If you want something that deep divergence, something that separate long time ago, this tend like very far away. You don't want something very fast, right? You want something to be a bit slower so you can still detect relationships. So you want the substitution rate to be very slow. And which structure is slowest here? Intron, exon, or spacer, right? You want exon. And in terms of mode of inheritance, you want something to be slow, therefore you probably don't want something that inherit from both sides, right? You want something that like uniparental, come from mom or dad alone, like not both. But on the opposite hand, if you want something recent divergent, something that's like super, like very recent, something just split up from each other, you want the evolution rate to be fast, and this probably go to intron or spacer, and possibly if you want something really crazy, you want biparental marker. If you want to study family, you want to know who your parents are, like very close, like the parents, who's a dad, who's a mom, then you need uniparental or biparental? You need biparental. And then, of course, the biparental usually fast evolve, and you probably need intron or spacer to do that. And finally, if you want to cover large group of organisms, you want to see the relationship between bacteria and eukaryote, like something really deep, something like go back in time. You want fast or slow? Slow, right? You want something that like super slow, like glacial pace. So this would be, you want a slow, and you probably want exon, and you want something that probably uniparental. So this is a very general criteria for choosing the right marker. It doesn't always work this way, but this is where to get started. And this usually pretty subjective, but this is how we can do it. And not hot to choose marker, is how to. Good God, autocorrect sucks. All right. So far so good. There might be some question on the quiz about this. So, for those of you who watch the video, this is where you want to pause and go to the quiz.